So still we are good morning, Assalamu alaikum, and good morning to everyone. I'm speaking on behalf of Parisa. She could not manage to come, and so that's why I will try to cover her part here. Of course, we work together, so and also we are husband wife, so I can take the opportunity actually to talk on her behalf. Now, where it goes, how would you strike? Wish me luck, that I don't have problem like you.
It's just the reverse.
Down one? Okay. Okay, that goes. I would like to start with Sweden because from Vietnam I would like to take you to Sweden, and why I will tell you. Because this university, SLU, we call it Swedish Agriculture University. This is located in a city or state called Uppsala. And in Uppsala, there are two universities. Uppsala University, people call it as a big brother university. And this young university, which is still ranking and considered rank, I call it 36 rank. So I feel very proud to be a part of this university. And our faculty of veterinary medicines and animal science is in the top 10 when it comes to the veterinary science. So that's why I try to tell that don't confuse with Uppsala University, that there are two universities in Uppsala, SLU as well as Uppsala University. So it's very important because I know that next time you meet me, you are from Uppsala University. We need to clear that out at the beginning of the presentation. Okay, so this is the faculty, and so this is the interior of the faculty. My office is somewhere here. And it's nice. We call it a colosseum. It's a round building, so it's really like a colosseum, but it's nice place. Of course, I'm showing you the pictures of the springtime, autumn time, where it looks very beautiful. But the reality is a bit different, and I will tell you why in a few seconds. So it's Uppsala. It's like 40 minutes by car from Stockholm, so it's not far, right? It's nice. But then also it's very nice when you have a temperature from -20 straight up to 33 actually. Actually 35 this summer. It went. So if you want to expose yourself to an extreme weather, you're most welcome to Sweden. Yeah. And I think Artemia's, this is a seminar on Artemia, we should be prepared to become an extremophilic like Artemia. And I think I am an extremophilic living for the past 5 years in Sweden. I'm turning into an extremophilic. This is the group. I have a reason why I have presented this group, and I will tell you why in a couple of minutes. So, two researchers. This is Parisa, who is in the steering committee, and then we have my colleagues, Alex, who is actually responsible for nutrition research. This is a department of animal nutrition and management. So he is the one who is heading the nutrition lab, the feed lab, and so on. Parisa is more responsible for the Artemia. And we are so lucky to have these two experts, the technical team. Actually, Astrid, she has a lot of biomedical research experience, but now she is also the lab director, I call it. And then we have the whole support team here with PhD students. These are the PhD students that I have based at the SLU. And obviously, you know this guy sitting somewhere there, Sarat, jointly supervised between Prof. Young and us, Parisa and me. And then we have this student from Norway, jointly supervised by Norway. And then this is an Indonesian student supervised together with Julie Kasari. And then we have quite a lot of master students, some in collaboration with Ghent University, Prof. Annelies here. And then we are also lucky to have a lot of visiting. And many of you know Julie, and these are also actually alumni of Ghent University. So it's nice to be connected in some way. At this slide, because I'm an alumni of Ghent University, and we are used to call internationalization. It's a difficult word for me. And when I try to put it in a picture, I feel so good to see that, there are a lot of students or researchers coming from different parts to SLU. This gives a lot of good satisfaction, because this is something that we have, I think everybody will agree, that we have learned from Professor Patrick Sormus. He likes that connection, right? Okay, the Artemia lab, Sweden and Artemia lab. There was no Artemia activities in Sweden 5 years ago. So we just started, thanks to Patrick, thanks to Peter Bussy, who supported a lot in this. So we built a small Artemia lab with the intention of using Artemia as a model organism for aquaculture research. I will stress this word again, model organism for carrying out aquaculture research. That was the idea. And everything went out very good. I have to personally thank Ling for this picture. I know you sent it to me. I was really looking for pictures, but you sent a very nice picture. So that's why you have your, I express my gratitude for that in person. Okay, so the responsible person is Parisa here. So we do a lot of works, and I will tell you in the next slide what we do here. The first thing, as I said before, the idea of having this lab is to use this as a model organisms, okay, for aquaculture research and beyond. Beyond, we talk about it a bit later. Next one is to use this as a feed for animal, aquatic animals. Again, an exclamatory mark. We talk about it later. And last but not the least, as food for human. But this is something that we have been discussing several times with Professor Sorglus, that this could be used as a food for human. So there we have the interest. I'm not doing any activity now, but I know that Hua has been presenting some slides on Artemia as a food or in the omelette, also Prof. Young. I think we have interest, so if there is any possible, I would be more than happy to learn more than to do, but so that is the third priority I have kept here. And I also the rationale behind these three reasons are that they are aligned with the aims and objectives of IAC. So it fits very well within the aims and objectives of IAC. Okay, why those three objectives? What was the rationale behind? The purpose behind three research lines, Artemia research line, is to address this problem, food security, to address the issue of food security. That is the overall aim of our research. And I don't need to explain this. There has been a plenary discussion. There was yesterday also people were talking about food security. We don't need to explain this to this audience that food security is an issue. But I would like to draw your attention that look at the number of people that are being undernourished, 690 people. And I can tell you, I think every time we are talking, there is number is, for sure it is increasing, if not decreasing. So it is a matter of concern. And there are a lot of factors and there are a lot of problem drivers, let me put it this way, that are causing this food insecurity problem. One is the feed. So if you wanted to grow fish or chickens or whatever farm animals, we need feed. We need soybean, be it fish meal, we need. And everybody in this house knows that this is not sustainable. Okay? So we agree on that. Next is disease. As Prof. Young also mentioned, Prof. Annelies also mentioned, disease is one of the limiting factors for sustainable production of any animal farming. Any animal, take any animal farming, we have issues with disease. So second, that's another thing is the habitat destructions. We are destroying our nature to culture fish, to culture animals. This is a big problem. We have limited space if you talk in the global world, in the larger scales. On top of that, we have the climate issue. We cannot deny that. And obviously, the other issues like socio-economic issues of the people. If you look at the people in Bangladesh, for example, there are lots of projects going on in the Cox's Bazar. Of course, thanks to Artemia, that there's a lot of changes happening there. But there are issues related to that. I believe that Artemia can play a big role there, if not directly, but at least indirectly, can play a big role in addressing those, I would say, global food security issues, let's put it this way. It can address those problems. So I will go back to this. It's obviously impossible to cover all the things, but at our lab, we try to pick up a few things to solve at least to certain level through using Artemia. And one of the things that we are trying to do is to screen feed ingredients. Please keep in mind feed ingredients. We will talk about it more in details in the next slides. The second we are trying to address is the disease issue using Artemia as a model. That has already been explained by our colleagues here. And last but not the least, Artemia can play a big role in understanding these ecological studies, climate change studies. So we will talk a little bit of suspense, but we will talk about it, right? So, circular economy. Many of you have already discussed, and Prof. Yong has already talked about this yesterday also, about the biomass or the waste that you have been using to feed Artemia. We are doing the same thing in Sweden. Sweden, they don't have a lot of food, but they are using so much of this circular economy approach to produce feed, to produce food. And I can tell you one thing, there is a mindset that we have to move from linear economy to circular economy. And everybody, including, I'm surprised, and now you will tell me I'm exaggerating, but I can tell you that even my daughter of 6, 7 years old, she even knows about circular and waste management. I'm surprised. At the age of 7 years old, I never thought about waste management, but they are putting it in the head of the children about waste management. Of course, she doesn't talk about the circular economy part, but she talks about waste management. And I'm happy that this is there in their mind. Okay, so what is happening is the waste to feed. So a lot of waste are being produced, and a lot of waste are being produced, especially in Sweden. This is a big forest industry. They use it for the furnitures, but the waste, what to do with the waste, right? So there is a heavy investment by the government to convert this waste into something called a resource. But in our case, the resource is the ingredients, and that is the microbial protein. So they are using so many different techniques to produce microbial protein and to use this microbial protein as a feed for fish, for poultry, and so on. And there come the role of Artemia. When I stepped in there, I was pitching the idea of that, why not we try to use Artemia to screen those things? And I was lucky enough to get to attract the attention of those researchers, and they believed in using Artemia as a model. So the idea behind was that we use this so-called waste resources, screen using Artemia, and then validate in the fish. So they are producing so many feed ingredients, selecting, testing one by one in fish, different fish. It's time consuming and it's also expensive. So why not we try a little bit simpler way? That's why and I can tell you that we were lucky, and we got funding also for that. And the reasoning I used to convince them first is obviously I always tell them it is cheaper, and that's how you get quickly attracted, and it's easy to handle and manipulate. That also, because if you use Artemia, you need less manpower compared to fish and all those things. So that was easy for me, especially think about Sweden when you have to pay a lot of for the technical team. Let's put it this way. Another thing is that also we will not go into details about the genome being sequenced, so we can understand how these microbial proteins or ingredients acts. What is the mechanisms? We can understand this. It's for academic people. It's very interesting for us, and that's why when we pitched the idea of using Artemia, they were very interested. And one of the most important thing that was triggering for them, and I will a little bit coming close to it, is dish. I used the word when I was there in Sweden. I was using the word no ethical concern when you use Artemia. But the students there say, "Are you sure that there is no ethical concern when you use Artemia? Don't you think that they have a pain or maybe I was finding it very difficult to convince. I didn't have data to prove them that the Artemia, they don't feel. I don't know exactly what happens. I know that there have been a lot of debate going on about the welfare of invertebrates. So I changed the word from no to less, so that at least I'm not questioned by them. And that's why I also get a little bit less. So if we use Artemia, we don't need to fill a bunch of paper to get the experimental work done. So that's also reduced a lot of time. And last but not the least, this is another reason why I could convince them that it falls somewhere within the 3 hours. It falls somewhere within the 3 hours. And that's why they got very interested. Obviously, we all agree with this, that use of Artemia as a model can fast track or fasten or speed up the screening process. Okay, so this is a little bit about the use of Artemia in screening and feed research. It fits very well. And I like to little bit give you a 2 quick. I can tell you these results are straight from the oven, so they have not been even processed, but I will still share with you. So we have 2 projects together with a consortium from the Nordic, but then also we have Canada and also Germany. So we got this project, and I was coordinating this project. It's a NordicFeed grant. No, we don't have the students here. It's NordicFeed grant. So what we did is that, just to reduce the time, so we can see these are all the way, this is the huge. Finland is the same. They have a lot of forest, they produce a lot of byproducts from the forest. And then we have the animal and the food waste. But this is the main thing, because that's a lot they produce. So by using all the techniques, we produce feed ingredients called the microbial ingredients or the microbial proteins. And then I use Artemia for screening this. And then eventually we prepared feed. My PhD students have just finished the experiment. And then the idea is to produce, some. Okay? And because it is a carbon neutral feed ingredients and all these things, you can sort of start getting this organic, organic is a big word, but sort of like an organic or natural status when you grow salmon out of these circular feeds. So I was using, this is the microbial protein. It is a fungal protein that we have. This is produced by a Finnish company, and this is recognized by the European Union to use. And in salmon industry like Biomar and all, they are using this. They are replacing 10% of their fish meal in the diet and replacing it with this one, to adjust the price and all these things. And also, I have to tell you this: when we were there in NordicFeed project, Norway has told, I don't know whether they can manage to reach it or not. By 2030, all the feed ingredients that at least in Norway has to be green. 2030 is a lot of pressure. I hope that we can manage, but we will see. So, and I think that was also the reason why we got this project, because it was fitting within that the pressure that was being built by the government. And I think when Norway starts something, Sweden and Finland, they're very quick to pick up the same sort of like the policy. Okay, so I will quickly show you the results here. So what I try to do is that when I told them that, look, you are using these pikiloproteins as a source of protein, but I think as it is a fungal protein, they might have some health benefit. Let me give a try with Artemia. And I did this, well, my student did this quick experiment, and you can see that, just to simplify it, this is the challenge, Artemia with challenge, and these are increased, so this is a control, this is the control, these are the challenge, and these are fed at different inclusion level. So what is interesting is that at this 5 percentage, you see that there is an increase in the survival. So these animals, Artemia, are being challenged. So there was an increase in the survival. When I presented this result, they find it very interesting. Companies, of course, they have a reason. So they told me something personally, which I can share with you, is that now I can increase the price of my product because of this result. So they are so happy to say that, because they see, before they were seeing the fungal protein as a feed ingredients, but now they see it as a functional ingredients. So you can imagine, and thanks to the Artemia, that gives you those sort of like a good feeling, right? But obviously, my student now, the PhD student, is trying to do more health work to really validate this, because we need to validate that. Okay, so just that, and now, Johannes, I think Annelies know, we are going to have a master students together with Annelies and Parisa and me, and again, this is in the framework of the Iraq, consortium. That's also a good sign that we are doing such type of work, where he will try to do a comparative study, checking which the microbial ingredients coming from different source, produced from different source, which one will act in what way, right? So we are really looking forward for the student. I think he will be around somewhere in February. So we were really excited to have him and do this work more on using Artemia as a model. Second is the health management. I will not go further on the health management, use of Artemia in the health management. Professor Annelies, Professor Yom has already explained this. But what I would like to draw your attention is that just to conclude, or as a home-taking message, that Artemia can be used as a model to do a lot of host microbial interaction studies. That's nice. Everybody in this room now is convinced, especially from the presentation of both of you. But what is important is that we are missing here. We have a very good survival response, growth response of the Artemia, but the mechanisms, how these things work, is a big a question mark for you, for us, in fact. So what we try to do is, at SLU, we are trying to develop the toolbox, the molecular toolbox that help us to understand the mechanisms, right? And I am already happy that Prof. Swilling has already done a lot of things. We have been having a lot of discussions the other day. And now that with the availability of the genome, and I didn't know that the Artemia, your species is also being sequenced, but that's good. But Franciscana, the genome is available now, and that is the time where we need to go step and go deeper on that, and to develop different toolbox. So we have been working quite a lot on developing single-cell omics studies, and also epigenomics marker. Parisa is heavily focusing on that. I know that some of the tools have been already developed, as Professor Young mentioned, like the RNAi technology, but then we need to focus more on the genomics and the omics, or the proteomics, genomics, epigenomics, proteomics, and so on. We need to develop that. But then for that, we need a good bioinformatician. That is very key for us. So we have, I have opened a position for a researcher in Artemia genomics, who can really go deeper on that, who understand these organisms, and I think this is a help for the consortium. It will be a big help for the consortium who are doing fundamental works also with Artemia. So I think by October, November, we will have this researcher employed. What we are also doing, again, another problem is that when we do this type of work, we use gene expression, we use the whole animal to do gene expressions. And this is another problem, especially when you work with vets, because the veterinary faculty that I work, they are all with vets. They always question me, Kartik, are you sure about the whole animal? We need to check the tissue specific. And that is why it got very triggered. So we are now spending a lot of time to understand, to isolate those certain tissues, and now we will I will try to say what you have been doing. So what we are trying to do now is to characterize the blood cells. But I'm so happy to see that you have been already doing it. But what we are trying to do is to characterize the blood cells of the Nopli. Whereas, if I'm understanding, you have done it in adults. So this is a big challenge, because the Nopli, collecting the hemolymph from the Nopli is a nightmare. But we were successful in collecting the hemolymph. That's at least some good sign from the Nopli. It is, I will completely give the credit to Parisa, because I have been just looking and she has been doing all those sort of things. And then we, after hemolymph collections, we try to use microscopic analysis to understand the cells, to characterize those cells. Again, that is very, very, very challenging, because there are so many cells coming from the body and tissue, it's very difficult to find it. But we are, I will tell you, we have a very, very experienced lab director who is helping us in that. I don't know if I can, there is a video here. I don't know, you can press it somewhere, there should be something. I don't know if I can do it use here. No? Can you go to the cursor somewhere here? Okay, but anyway, this is okay, there it goes. So this is a decapsulated cyst. It's a 3D image, and we have used confocal microscopy to understand. And these are with pathogens and without pathogens. I'm showing you without the pathogens. The thing is that we are trying to use confocal microscopy to see the interactions. It was not very easy because of this, a lot of interference of other cells. Very difficult. But I'm sure if we have done it with adults, I'm sure we will also be able to do with the Nopli. And anyway, this image we did it, but then I want to draw your attention to this image. These are all the blood cells actually. We don't know which one is blood, which one is granular site, which is agranular site. I have no idea. We need to characterize this. And as because you see, when we try to see the image, we have a lot of these algae also coming in between. So it's a very difficult. But I will keep at least one thing we could succeed is that we have used a protocol to get it more cleaner version. So this is a bit cleaner. At least one step we have moved further to from this messy world we came to the cleaner world. So we can screen them, we can find the size. But what are they? That needs to be checked. And that's where we are now collaborating, and we were so lucky, and I think Prof. Sorglus here knows him very well. We were very lucky to have Professor Eric Cox, and this is again with Ghent University, to have him as a visiting professor in our department, who is sitting next to us now, and he is helping us because he has an extremely good experience with cell culture and all those things. So he's helping us to at least characterize those cells, and Parisa has been to his lab. They have developed different cell culture techniques, and we are now trying to collaborate with Eric to use his experience from pig also to see if we can do the same thing with Artemia. But I know it's a difficult road, but we will see how much we can go. And I will have to, of course, thanks to our Astrid, the lab director, who has a lot of experience with cell culture technique. Yes, this is something very interesting when it comes to Artemia ecological studies, and I even call it like Artemia cyst biology project, whatever you call it. And I know we have quite a lot of discussion when we were in Terengganu last time. We had some discussions with Dr. Hoa, Professor Sorglus, with Prof. Young about using certain mechanistics tools or molecular tools to study the cyst biology. I have some results, so I will just quickly explain to this. I hope I can explain because this is mostly the work of Parisa. So we have got the cyst from Winchao, also from Urmia, from also Sunny. We have got the cyst from different places, especially also from the United States, to do some cyst biological work. And I'm a little bit happy to share with you that we got some small, it's called seed funding, to use this from the university to develop this method on Artemia. Because this method has been developed for other organisms, not for Artemia. So we are having a hard time to optimize it in Artemia, but I'm sure again we will be able to do that. So the protocol is based on certain techniques where you use the Artemia sample to study both the genome and the methylome at the same time on the same sample. So normally we have this hi-fi called PacBio where you go for whole genomic sequencing, or you have the methylome sequencing. But this protocol allows us to do the two things together on the same time. And this means this is much cheaper and easier and realistic than the other two. Well, of course, I cannot ignore the other two. It has its own merits. But we prefer to do something where we can do both things together. And that makes the price dramatically less. So these are very, as I say, straight from the oven. These are the two results. We have been trying to see the frequency of the methylated sites. It's more the deep methylations in the Artemia samples. It took a long time to digest the samples. It's very difficult. to do that. We have either a DNA damage and I think, again, I cannot, maybe this afternoon, Parisa can give more explanation to that, but I know that she has been coming home with always with a frustrating face, and I had to tolerate her. So I will not speak too much about it. So this afternoon she can give more information. So the idea is to developing biomarkers for special or cyst with certain characteristics, which might help us in our inoculation missions in a certain region. That's the whole idea of the project. So let's see where we are, where we reach actually. I told you the intention of when we started Artemia, the intention was to use Artemia as a biology or model organisms for aquaculture research. But if we develop these tools and if we join forces, and now I'm very convinced about with this team, consortium, that we can present this Artemia as a model for biological research, more in a concrete way. So with this, I would say thank you very much. Terima kasih.
Thank you.